Okay, so let's talk about neurodevelopment or how the nervous system actually develops, how it comes into being when we are little embryos and fetuses. So the outline for this section is we're going to start with the zygote and how it gets to the trilaminar disc. Then we're going to talk about formation of the neural tube, which then differentiates into a 3-vesicle stage and then a 5-vesicle stage and then the mature stage to create a brain that's something that you would recognize. So let's start with the zygote. So in the beginning, right, we all started from a sperm and an egg. So a sperm is the gamete that has 23 chromosomes that usually comes from a male-bodied person, and then the egg or oocyte with 23 chromosomes from the egg donor or biological mother. And then those two cells fuse and their chromosomes come together to form 46 chromosomes total. And that fertilized egg with the 46 chromosomes is called a zygote, which I think is an awesome word. So we see here on the left that zygote, that fertilized egg, that is what we all start as, is one single cell. They're showing you those two nuclei, one from the sperm, one from the egg, fusing and coming together. That single cell then with its 46 chromosomes is then going to make copies of itself. So it starts making identical copies of itself until you get more and more and more cells. So that's what we're seeing as we kind of like go through the days after fertilization here. And then but initially, all of these cells are exactly identical to each other, but then something really exciting happens. It says here around day 5, it's usually around day 6 to 8, but some of the cells become this outer ring of cells here. You can kind of see it between those two red lines. I'm not doing a great job. And they're calling it the troctoectoderm. We usually call it the trophoblast. Don't worry, you don't need to know it. And then, so some cells become that. And then another group of cells says, you know what, we're going to actually become this group of cells here, which is called the inner cell mass. So this is a really exciting moment for biologists because this is the first time that those cells, which were all identical to each other, actually differentiate, become different for the first time ever. Some are going to be part of that trophoblast, that outer shell of cells, and then some are going to be part of that inner cell mass, that little clump of cells in the middle. Now that clump of cells in the middle is what's going to become the embryo or the future baby. The trophoblast around the outside is going to become the amniotic sac and a whole bunch of other stuff. So we're going to focus in on that inner cell mass and what happens with that. Okay, so this is a similar graphic. So we had this ball of undifferentiated cells over here, and then some of them become this outer ring of cells, and others of them become this inner cell mass, this clump of cells here. So that inner cell mass is then going to form a disc. So if I just take this section here, like that, right? So this is 3-dimensional. This is like a sphere, right? That is going to be an oval disc, a flat little disc. It's the coolest, strangest looking thing. Let me show you. So this is a micrograph, a microscopic picture of a pig embryo on day 12 when it is an embryonic disc. So pigs, chicks, rats, elephants, we all do this. We all follow this same progression of development. So we all form an embryonic disc. So you can see here is that side view, kind of like what we saw on the previous diagram. And here is like a top view, which is what we're seeing in the micrograph. So we're all this disc, right? So we're a disc, and then if you look at it from the side, we're going to start to see that it's going to form some different layers. So we start as an embryonic disc, and then that disc is going to form 3 distinct layers. So you can see in this last image here, where's my thing? Okay, we've got a blue layer of cells, we've got a yellow, layer of cells, and they're starting to make a little orange layer of cells. Now they're not really those colors, those are just the colors that are commonly used in diagrams to help us visually distinguish things from each other. So here again we are inside that bubble. This is our disc here. And if we zoom in, we're going to see we start off with just these blue cells. But then, growing down from the middle and spreading out, we make these yellow cells. Now, the blue cells are going to become something that we call ectoderm. The yellow cells are endoderm. So we start with those. We start with the blue ones, then we make the yellow ones. They aren't really those colors. And then third and finally, we make these red ones, which are the mesoderm. So we're going to form a disc with 3 layers. That's where the word trilaminar comes from. Tri is in 3, like tricycle has 3 wheels. Laminar, like laminations, those are layers. This is what that looks like on an electron micrograph. So super, super powerful microscope. And you can see they've got the ectoderm is up here. That doesn't show up very well. Endoderm is down here, and mesoderm is in the middle. Kind of cool. So that trilaminar disc, that 3-layered disc in the middle of what was the blastocyst, right, is now going to become everything. It has to become everything of the embryo and future fetus or baby. So in broad strokes, this is an overgeneralization, but the ectoderm, that first blue layer, is going to form the skin and the nervous system. So that's why we're talking about it. And then the mesoderm, that middle orangish or red layer, is going to form your muscles, your skeleton, your reproductive organs, your kidneys. And then the yellow endoderm is going to form kind of your innermost parts of everything, the linings of your intestinal tract, the lining of your respiratory tract. A lot of your squishy bits that are on the inside are going to be formed by the endoderm. Okay, so that was from zygote to blastocyst to inner cell mass to embryonic disc to 3-layered embryonic disc with ectoderm, mesoderm, and endoderm. Now we're going to focus in on how that ectoderm is going to actually become the nervous system. And the first step in that is something called neurulation, which refers to formation of something called the neural tube. So now we're going to talk about how we form the neural tube. So first off, a groove is going to form. So here's my trilaminar disc, and a groove is going to form all along the center. That's going to be called the neural groove. All right, now watch. That groove forms, and then the edges hump up together, fuse, and then that tube comes down in between. So we're going to watch it again. Those edges fold up, they fuse together, and then we pinch off a tube that then goes in the inside. So gifts can be annoying. So let's look at it. Okay, so again, same thing. Here we are, 3-layered disc. Okay, we're going to start to form a little groove here. Those edges of the groove are going to heap up. They're going to fuse together and pinch off. So then we're going to have a tube in the middle that's completely separate. Now it's completely separated from the rest of the ectoderm. So it's now entirely inside of what is going to become the embryo. Now that mesoderm, that pink stuff around it, is actually going to form the spine. So when we learn about the spinal cord and we see how it is entirely within the spinal column or the vertebrae, you can kind of now see how that happens, which is exciting. Okay, this is another electron micrograph looking at an embryo, not a human embryo, probably a mouse actually. So you can see that groove starting to form here. And then here, those edges that are going to hump up and pinch off together around that neural tube, which is cool. And then you see that the rest of the ectoderm is going to form the skin. This is the same process, so neurulation. This is the diagram from the Hedges, the Valerie Hedges textbook. I don't like that they don't use the same colors that everybody uses, but that's okay. And it's because they want to show you the different parts of the ectoderm, some that become the nervous system and some that stay as the skin. So you can see we start off here with just a little beginning of a groove here, and then we heap up these folds on either side. And what I like about this diagram is you can see a top view, right? Because this is running down the whole length of that embryonic disc. And then eventually that tube is entirely within the embryo, right? So now we don't see any of that neural tube because it all zips up and it gets closed off inside of the body. So what if it doesn't zip up properly, right? So anytime something can go wrong, it usually will, at least on occasion. So there are some medical conditions that happen where that doesn't zip up properly, and parts of the neural tube are still outside of the body. They're not underneath the skin the way we're seeing here. So if you've ever heard of neural tube defects, that's what that is, right? Now you know what the neural tube is, now you know a defect of that. So here is an example of one type of neural tube defect called spina bifida, where it fails to zip up way down here at the bottom of the spine. Now there are varieties of severity. There's different degrees of severity of spina bifida, but of course the most severe one is where you have a little sac of membrane with spinal cord and nerves and things all hanging outside of the body. I'll show you a picture of that. So you can see that. So that red mass at that baby's lower back there is actually got spinal cord and nerves in it. As you can imagine, that's not where spinal cord and nerves belong, so this can be devastating. This baby obviously is in the ICU. They have lots of tubes and monitors on them. They are going to have surgery to put this inside the body where it belongs. Now, neural tube defects can happen in other places on the neural tube as well. If it happens way up top, the part that's going to form the brain, then you end up with a brain that is open. And so those are really severe, so those babies often don't survive even all the way to birth. So, okay, so we have to ask ourselves, when during pregnancy do neural tube defects occur? And trilaminar disc and neurulation is all happening around the first 5 to 8 weeks of pregnancy. So this is happening really, really early in the pregnancy. And I think I forgot to put a diagram in here. So you're just going to have to remember that I said that. Yep. All right. So, but there are things we can do to help decrease the likelihood that somebody will have a neural tube defect. So it's interesting that folic acid supplementation significantly decreases the risk. Now, folic acid is a naturally occurring vitamin that is present in leafy green foods, a lot of legumes, some whole grains, but most people don't get a diet sufficiently high enough in folic acid to reliably decrease their risk of neural tube defects. So for that reason, one of the main ingredients in prenatal vitamins, those vitamins that you take when you're pregnant, is actually folic acid to help prevent this from happening. But now remember, this happens really early in pregnancy. It's just this big, right? It's just the first 6 weeks or so, and a lot of people don't even realize they're pregnant that early in the pregnancy. So for that reason, most medical organizations recommend that anybody who's of an age where they could potentially get pregnant should actually be taking a folic acid supplement. Now, of course, a lot of people aren't doing that. And so to help also decrease the rate of neural tube defects, many cereals and grains in the United States are fortified. They have a small amount of folic acid added to them. As a culture, we don't tend to eat a lot of leafy greens, and so giving us a little bit of extra of that vitamin in our breakfast is probably a good idea. So I looked at my cereal box from this morning. I had strawberry Cheerios. And if you look down on the labels, you will see that it is supplemented with folate or folic acid. You can see it right there. Now, 20% of the RDA, right? So it's not a super high dose. That's about 1/10 of the dose that is recommended for pregnant or potentially pregnant people. So it would be hard to overdose with cereal. You'd have to eat a lot of bowls of cereal. But just this little bit can actually help. So this is data looking at Canada, actually, because it was a nice article in the New England Journal of Medicine. I liked that they showed this particular graph. So what they're showing you here is the rate per 1,000 births of neural tube defects in Canada. Now, they've broken them out by different types of neural tube defects. Spina bifida, which is the one that you saw the example of, is the one in the orange. Anencephaly is one where the upper part of the brain doesn't form at all because it doesn't fuse together. That's one of those really severe ones. That is the one in purple. And even if you just kind of sit back a little bit and take a look, I think you can notice that all of a sudden things started to get a lot lower. And in fact, 1998 is the year that Canada started fortifying a lot of their cereals and grains and breads and things with folic acid. So this is a public health intervention, right? This is just kind of helping bring the baseline up for everyone with significant health benefits. All right, so that was neurulation, or formation of the neural tube. Now we have to turn that neural tube into a nervous system, or the parts of the nervous system that we're going to talk about next. So what we're looking at here is that embryonic disc, and this whole thing is the embryonic disc, and this whole bit here is the neural tube. Now you notice that this anterior or front part of the neural tube has kind of swollen out like you're blowing up a balloon, right? The tube has dilated, and it actually forms three distinct regions. It forms the prosencephalon, otherwise known as the forebrain, the mesencephalon, otherwise known as the midbrain, and the rhombencephalon, otherwise known as the hindbrain. I guess somebody thought it looked like a rhombus, which is a four-sided shape. I don't know. So that anterior neural tube, the top part, the part most towards what's going to become the head, right? We're still just a disc at this point, but the part that's going to become the head is going to kind of balloon out. It's still just filled with fluid, which is pretty crazy. And then it's going to start to form kind of like three distinct little chambers, the prosencephalon, mesencephalon, and rhombencephalon. And those three little ballooned out parts of the tube are going to end up forming the brain. The rest of the neural tube is going to end up forming the spinal cord. Okay, so let's focus in on these three that are then going to form the brain. How do they do that? Well, they go from the 3-vesicle stage to the 5-vesicle stage. So that prosencephalon, that forebrain, is going to further subdivide into two little bubbles, the telencephalon and diencephalon. Mesencephalon stays the same, and rhombencephalon also subdivides into two little bubbles, the metencephalon and the myelencephalon. This is what that looks like. So from a lateral view as well as kind of a head-on view, a frontal view, which I appreciate. So different colors, again, my apologies. So here we have the forebrain, the midbrain, and the hindbrain, prosencephalon, mesencephalon, and rhombencephalon. And then they're going to further differentiate. And notice here we've got the eye cup. You know how sometimes you see those billboards and it says, my eye is formed at 14 days or something. It's not really an eye, it's an eye cup. It will eventually form into the structures of the eye, but anyway. So at about 5 weeks after development, this is what the brain looks like. It is a hollow tube filled with fluid, right? Not very impressive, but it's going to become all of the things that you are familiar with. So let's kind of take a look at that. All right, so that forebrain, the topmost part, the superiormost part that forms the telencephalon and diencephalon, is going to become the cerebral cortex, that outer wrinkly part of the brain that is the biggest part of the human brain, as well as the basal ganglia, which are within the cerebrum, and you may not have heard about them before. So we're going to learn about them in this class. And then the forebrain also forms the diencephalon, which is that very, very middle in the center part of your brain that contains the thalamus and the hypothalamus. The mesencephalon just stays the midbrain. And then the rhombencephalon, or the hindbrain, becomes the pons, the cerebellum, and the medulla oblongata. Now this is kind of interesting because we're organizing it based on which bubble of the neural tube it developed from. But if you're thinking to what you learned in anatomy and physiology or human biology, you're like, wait, there's cerebrum, cerebellum, and brainstem. And the brainstem has the midbrain, the pons, and the medulla oblongata. And that is correct in terms of its anatomic location, but in terms of its development and where it came from, right? So the midbrain actually came from a different place. So kind of cool. All right, so let's take a look at the mature brain, something that is going to look familiar to you, and just remember where it all came from. So that prosencephalon, that forebrain, differentiated into telencephalon and diencephalon, and the cerebral cortex came from the telencephalon. So cerebral cortex, then you probably remember, I hope you remember, has those 4 different lobes. We have the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. So that comes from the forebrain or prosencephalon. Here are those basal ganglia that I mentioned, and they're kind of deep inside the cerebrum, which is why we often don't talk about them in anatomy and physiology, but we'll get to learn about them in this class. They also came from the telencephalon, which came from the prosencephalon, and they're these little kind of islands of specialized cells found deep within the cerebrum. And then the other part of the prosencephalon became the diencephalon, which is that middle part of the brain, which contains the thalamus and the hypothalamus. Mesencephalon became the midbrain, right? Which is, it's kind of hard to see here. I tried to gray everything else out, but the midbrain is this brown section right here. So there's the midbrain. The rhombencephalon, the hindbrain, you can think of this as the most primitive part of the brain. So these are things that are absolutely crucial for life support, which is why medulla oblongata, of course, is my favorite part of the brain because it keeps me breathing. Pons, which is a really important relay station, and cerebellum all came from the rhombencephalon. Okay, so this was a lot of crazy words that you've probably never heard about before. So what am I actually expecting you to know and retain? A couple of things. I want you to understand those 3 initial bubbles or vesicles. So prosencephalon, which is the forebrain, mesencephalon, which is the midbrain, and rhombencephalon, which is the hindbrain. And then you can see where they all, where the structures of the brain we're going to learn about in this class are all coming from. So I want you to know all the things in this table. The other thing that I want you to know is, in general, the higher up you are on this table, the more complex the functions that are occurring. Whereas as you move down, these are kind of more basic brain functions, things that just keep your body functioning, right? So basic kind of life support, making sure you maintain homeostasis, some goal-directed behavior like eating, and breathing and maintaining your blood pressure, all of those things, and being able to move, at least in terms of reflexes. So those things are kind of the more primitive functions of the brain, and then the more complicated things like writing poetry or studying for a neuroscience exam, that's going to be up in the forebrain. So in summary, what did we talk about in this little lecture? We talked about how the zygote, the fertilized egg, then becomes a blastocyst with an inner cell mass. That inner cell mass is going to become the embryo, and it does so first by becoming that disc that forms 3 layers: ectoderm, mesoderm, and endoderm. And a part of that ectoderm is going to fold in and form the neural tube, which will normally come completely within the body underneath the skin. But if there's a failure of that process to occur normally, those are neural tube defects, and we can reduce the risk of neural tube defects with folic acid supplementation. Now, you may have noticed they don't go to 0, right? There are other things at play. There's genetic factors, there's other exposures, there's lots of things that can combine to cause a problem here, but making sure you have enough folic acid can significantly decrease the likelihood. Then that neural tube, which really just starts off as just a plain old tube, starts kind of bulging out anteriorly into those 3 different vesicles that are still just filled with fluid at this point. So the prosencephalon or forebrain, which will eventually become the cerebral cortex, the basal ganglia, and the diencephalon, which is thalamus and hypothalamus. The mesencephalon, which becomes the midbrain, love that one, easy, and the rhombencephalon, which will become the cerebellum, the pons, and the medulla oblongata. So that's it. That's where you got your brain. All right, see you soon.